Okay, so now that you have seen the different types of trees, additive, cladogram, ultrametric, and I told you that we count the mutations. So now I'm going to show you what the computer is doing to produce those trees. And I'm going to show you the first approach for historical reasons. And I'm going to tell you some variations, but I wanted to be aware that since then there are lots of improvements and ways of how we should account for differences. So you don't have to know all of it. You just need to know that the way the computer produces this tree is based on some counts and a lot of statistics. And I put a little story in the list of slides in case you're curious, and then you could see. So people make careers doing phylogenetics, and then when they disagree, it can get really heated. So it's kind of fun to see the human side of this. So in this lecture, you'll see how the phylogenetic trees are built. I will tell you already that it's a tedious process, similar to the traversing the matrix for dynamic programming. And that is why we delegate this task to computers. I will tell you what parts of this I wanted to know in terms of being able to answer questions in homework and exam. So you're going to know that there are different clustering methods, which is how do we group things. There are different ways of counting distances. So distances are the differences between the entities you're comparing. And I'm going to have you do an exercise, which is calculating p distances. So it's how different they are in proportion to how long the whole sequence is. And I'll give you an example that is a simplified version. You know, it's a toy example, so it's not a really long sequence. It's just to convey the idea of how it's done. And here when I say beware, I just wanted to know that there are other ways of calculating the genetic distance. So Jukes Cantor were mathematicians that said, well, I think we should make these corrections to this too. And you don't have to understand the corrections. And if you're not a math major, you might go like, oh, when I show that slide, that's okay. You know, even math majors go, oh, but then they actually go through it and they go like, oh, I see. But for me, I just want to shock you a little bit and just tell you, look, there's all sorts of adjustments we could do to this, but we're just going to do the simplest one. And like I said, this to tell you, like, how do these processes start? They start simple and then people start thinking, but we should account for other things too. Then they become more complex. So there are different ways of clustering, as I said up here. We're doing this one, EPGMA. It's the easiest. It's not as good as these other two, but it's the first one. So historically, this was the first way that people came up with how to group, how to cluster the entities. I'll show you examples of these, but so just so that you have some idea, but we're not actually going to calculate them. And the reason why I show you these names is that when you are running software, if you run into these names, I want you to know like, oh, different ways of clustering it. So you don't freak out like, oh, I have no idea what this is talking about. Okay, you already know about tree topology by now. And then there is this last thing that is an exercise using the UPGMA clustering method, which will probably continue Monday, but I will stop probably when you are starting this, but it would be good for you to give a stab at it, because when you're in class, that's what I do. I let you guys do this, and then I walk around and I help you, and then I show the answer. The answer for the PGMA exercise is in this slide sets. Okay, so when we are doing phylogenetics, we are again looking at, we're trying to represent evolution. So this closely related to this, and then this is the last related to this animal, so some plant. You already know that by now, right? So just to review, leaves. Like, Dr.T, these are not leaves. Well, we call them leaves because this is a tree. Or we call them terminal nodes. Then these guys are internal nodes. So they're the ancestors to these leaves. Internal node, ancestor to all these three. That would be our root. This is an edge or branch. And if it has a scale, then, or in this case it has time, then it represents how many mutations occurred between an ancestor and then this other ancestor. All right, so how do we build that phylogenetic tree? It's not magic. So you align the sequences. As you guys have seen, if you're doing a multiple sequence alignment, then you are using a global alignment tool. So you align all of the sequences. We're going to score their differences, and then we're going to make groups that are based on the differences. So whenever we talk about genetic relatedness, we like to think how similar they are, but the way we calculate how similar they are is by how dissimilar they are. Okay, so it sounds confusing, but the calculation is what is different between them. So we make a matrix called the distance matrix. So this is from this book. Some of you asked for references. That's the book I used for my master's in bioinformatics. But the reason why I don't actually use this book is because when I was doing this, I already had a PhD in genomics, and the language it's a little difficult to understand. Like I felt I understood, struggling a little bit, but mostly because I already had a lot of background. But sometimes I'm like, oh, it has a good figure, so I'm going to use the figure from the book. So this is basically to show you different ways of building the phylogenetic trees. We're going to use p distances. And the models of evolution, instead of using p distance, they will use this corrections. So I will show you Jukes Cantor, and I haven't told you about transitions and transversions. So you don't actually have to do all this. Purpose of this figure is to show to you some background on what goes into building a phylogenetic tree. So, and then the clustering methods, I told you that I'm going to show you examples of these three, but you're really only going to do the UPGMA. All right, so the stuff in gray is all the different ways that we can assign a numeric value for the genetic distances. And these is how are we going to group them? All right, this is the slide that I told you we're going to go, So this part is easy. The Hamming distance is just the count. So here are the different ways of calculating distance. The Hamming distance is the count. You just count, you align things, and then you say how many amino acids or nucleotides are different. So it's also called the not corrected or non-corrected number of mutations. And then somebody said, well, but if one string is longer than the other, then we need to account for the length of the strings. So that's called the p distance. So it's the proportion of the nucleotide or amino acids, if you compare amino acids, sites where the two sequences are different. And then the Jukes Cantor, I told you these guys are mathematicians. They're like, no, I think we should also establish these corrections. Okay, sure. And then Tajima Nain, they said, oh, I think these should also be corrections we make. So you're like, sure. So if you are working on this, who knows, maybe some of you would go into phylogenetics, then you are like, oh, is it Tajima Nain? Then you know it's counting the differences in a different way than what we're gonna do. Okay, so far so good. Are you properly scared? All right, so now relax because now relaxed because we're just going to do the p distance. And we're even going to do like a very easy version. That is, we're just going to take length of a sequence, not even all of the sequences. So here I'm showing you the Hamming distance. So it starts with, I have 4 DNA sequences. And then I align them. See how they're all aligned? They're like, oh, okay, there's an indel here. And then what I do is I compare 2 by 2. Remember how I did that figure with you guys before? The way we compare a bunch of things is we just pick 2 initially. So here we are going to compare A with B, A with C, A with D, B with C, B with D. We're going to do that in the next slide. On this slide, I'm just showing you what a Hamming distance is. So if you're counting the distance between A and B, then I'm copying this over here, and then I put in red the stuff that is different, and then I'll go, okay, this is different. 1, 2, 3, there's an indel, 4, 5, 6. The Hamming distance between A and B is 6. And then the Hamming distance between B and D, I just copied over B and D and I put them over here. The reason why I did that is to show to you that, okay, this gap doesn't actually exist in the sequence, right? That gap was just put so that C and A could align with B and D. So when I'm comparing B and D, I don't even need that gap there. So that is not a difference. And so the difference between those, which is the Hamming distance, is 2. So one nucleotide change, 2 nucleotide changes. That's all. But you need to learn Hamming distance to see, okay, how am I going to count the p distance? Okay, the p distance takes into account how long this string is. So here's the p distance between A and B. And so that would be, okay, there's 1, 2, 3, 4, 5, 6. So that's the Hamming distance. But then I count the length of the sequence. And I elected to get the sequence that is longer because I don't want to underestimate the difference. So I'm just doing 6 over 10 because there are 10 nucleotides here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Good. I didn't make a mistake there. Good. So that's the p distance. And then when we are making a tree, we do that for all of them. So as in, this was the p distance between A and B. How about the p distance between A and C? So the way you do that is you make a matrix. This is what I told you is called the distance matrix. And you fill out only half of the matrix, right? Because the other half will be a mirror image of, well, not quite half, right? So there is this diagonal here, which is, how different is A from A? The answer here is 0, right? If you compare you to yourself right now in this same second, so this sequence to itself, there's no difference, so 0. So the diagonal will be zeros. And then you fill out either the bottom or the top, I don't care. But this is the part where you're going to spend, let's say, 2 minutes, maybe 3. So it's now 10:39. I'll give you until 10:42 to fill out the rest. Oh, yeah, 10:42. See you at 10:42.